Hi, my name is Sahib, and this is my Biology of Cancer presentation number 2. The title of the paper I'm presenting is Resveratrol enhances the efficacy of sorafenib mediated apoptosis in human breast cancer MCF7 cells through ROS, cell cycle inhibition, caspase 3, and PARP cleavage. The cancer this paper examines is breast cancer, specifically MCF7 breast cancer cells, which are used in the analysis. So the reason, breast cancer is one of the main causes of cancer-related deaths among women worldwide, so it's definitely an important area to explore. And the main reason this study was done is because many different treatments of breast cancer are highly toxic, and sometimes there's drug resistance as well and unwanted side effects. So there was a need for a novel therapeutic approach with better efficacy to treat breast cancer. So that's why they tried out this method where they took a proven anti-cancer drug, sorafenib, which is used in many cancers and breast cancer, and they tried to increase its efficacy using a natural antioxidant or a natural occurring substance, resveratrol in this case. Well, we'll talk more about that. But in any case, they did conclude that a combination of these two substances showed a higher cytotoxicity or higher apoptosis rate on MCF7 cells than individually treating them with each of these compounds. And they observed several proteins being up or down regulated as a result, and we'll examine them in the paper. So first, let's discuss sorafenib. Sorafenib is a multi-kinase inhibitor. So the paper mentions that it inhibits two kinases in the RAF-MEK-ERK pathway, which we learned about in our last unit. So RAF is a kinase, so sorafenib inhibits it, and you can see it right here. And it also inhibits receptor tyrosine kinases like VEGFR1, 2, and the whole VEGFR family, or vascular endothelial growth factor receptor, which is receptor tyrosine kinase and is inhibited, as you can see right here. So it inhibits RAF and RTKs to inhibit the ERK pathway in order to slow tumor progression, as the ERK pathway is related to cell growth, so it would grow the tumor more, but in this case sorafenib inhibits it. And it also has anti-apoptotic properties as well. Next, we have the substance resveratrol. So resveratrol is a natural polyphenol compound found in berries, fruits, etc. It's also an antioxidant. It's been extensively studied in previous trials, and it's found to have potent anti-tumor activity, as well as chemopreventative activities such as activation of caspase 9, which furthermore activates a caspase cascade for apoptosis, but we'll discuss that later. So they're trying to determine the action of these two drugs, these two substances together, and their synergistic effects on the breast cancer. So yeah, we'll move on to the first figure right over here that we'll be examining. So figure number 4 right here, sorry. Figure number 4, so there's two parts of this figure, A and B, which are bunched up together. So A and B are basically a fluorescence test or fluorescence assay, and it's measuring, the fluorescence they're measuring is of a dye, a fluorescent probe. It's called H2DCFDA. And what this does is it detects things or something called ROS, or reactive oxygen species, intracellular reactive oxygen species. So we haven't learned much about it in class, but the basic overview, they're just molecules, oxygen-containing molecules inside the cell, and the paper states that cancer cells have a much higher level of ROS than regular cells, and higher levels of ROS leads to apoptosis, or the beginning of apoptosis. And our ROS is also associated with p53 activation, which we'll learn about in these next figures right here. So basically, the dye, the H2DCFDA, which is used as a fluorescent dye or probe in this, the fluorescence intensity of this indicates the level of ROS. So higher intensity, the higher level of ROS you'll get. So over here we see that the control has relatively low levels of fluorescence, not many lights, but we see the sorafenib group, or the cells that were treated with sorafenib over here, and the cells treated with our resveratrol have a little higher fluorescence. But the most fluorescence by far is in the group that was treated with both sorafenib and resveratrol. So this shows that both when acting together, sorafenib and resveratrol lead to much higher levels of ROS, as we can see through the higher fluorescence intensity. This basically means that there would be higher p53 activation. Like we said, ROS is associated with p53 activation, and it plays a pivotal role in apoptosis. So this could indicate that, both of these substances together have a higher, induce a higher apoptosis rate in the breast cancer cells. But we can see it more clearly over here in the Western blot in figures C and D. So B is basically just a graph that shows the levels of the fluorescence intensity. So it's basically the same thing. Similar for D for the Western blot C. So over here we can see that they're examining three proteins which we find in the cell, and these proteins are actually in a pathway, and they have specific roles. So first we have p53 that we said ROS played a role in activating as well. So, and we have BAX and BCL2. So p53 is a transcription factor for BAX, and it's also a tumor suppressor gene. There's also evidence that p53 acts as a repressor for the BCL2 gene, but still that's not confirmed, so we won't go much into that. But we can see over here in this basic diagram, if we're not looking at this part, we're just looking at this part right here. So we see that p53, when we have higher levels of p53, the transcription factor, there's more production of BAX, which is a protein which is on the surface of mitochondria, and it's a pro-apoptotic protein. So the action of BAX is basically that it opens a channel on the mitochondrial surface called VDAC. So VDAC is a protein channel that allows this substance, cytochrome C, to exit the mitochondria and combine with APAP1. We'll talk about the next steps after this for the next figure. But so that's what BAX does. It's a pro-apoptotic protein that allows cytochrome C to exit the cell, whereas BCL2 is an anti-apoptotic protein on the surface of mitochondria as well. It has an opposite effect to what BAX does. It makes the VDAC remain closed and doesn't allow the cytochrome C to exit the cell. So therefore, it can't combine with the APAP1 and form the apoptosome. So we see that, and we see this inhibition sign right here. So what does the Western blot say? The Western blot, it basically shows that p53 is shown in much higher amounts. There's some amounts of p53 when only sorafenib and only resveratrol are added. However, when both of them are added together, there's much higher expression of p53. It's the same thing with BAX. There's a much higher expression of BAX when both sorafenib and resveratrol are added together, as opposed to just sorafenib or just resveratrol alone, or the control, nothing is added. So p53 and BAX are upregulated greatly by the combination of sorafenib and resveratrol. However, BCL2 has the opposite effect, as we can see here on the Western blot and on the graph. Instead, BCL2 is inhibited inhibited greatly when both sorafenib and resveratrol are added together. They're inhibited even more than when just sorafenib and resveratrol is added. So this shows that it's upregulating this pathway, kind of. You see p53 increased when resveratrol, the expression is increased when resveratrol and sorafenib are added together. Bax is also increased, so you'd have more cytochrome C exiting the VDAC because the VDAC's open, and because there's less BCL2 because it was downregulated more when sorafenib and resveratrol were together, you'd have less inhibition of apoptosis, basically. So as we move on to the next figure over here, figure number 5. So figure number 5A and B, they're joined together. This is just a graph of figure A. This is again a fluorescence assay, but in this case, it's measuring something called the mitochondrial membrane potential. Again, we didn't learn much about this in class, but the basic idea is, according to the article, the collapse of the mitochondrial membrane potential. So as it goes down, it's been associated with the initiation of the caspase cascade, which we will learn about in this Western blot right here. So in this case, what the fluorescence shows, the fluorescence is actually a rhodamine 123 fluorescence dye, which measured the mitochondrial membrane potential. So once this dye was added, we could measure how high, and we measure the fluorescence intensity of this dye to see how high the membrane potential is. So the more brightness, as we see over here in the control, the more membrane potential. So we can see over here that there's a very high fluorescence, so a very high mitochondrial membrane potential in the control. Somewhat less in the resveratrol and just the sorafenib cells, where just resveratrol or just sorafenib was added. But in the combination group, we see that oh my God, it's almost non-existent. It's greatly reduced. So it's almost like the article said, a collapse of the mitochondrial membrane potential, which is apparently associated with the initiation of the caspase cascade, which we will see over here. So there are 4 proteins analyzed in this Western blot over here, and they're more related to the apoptosis, and they're actually still related to the proteins we saw last, because so APAF1 is a protein over here, as we can see. It actually combines with cytochrome C when it's coming out. 7 molecules of cytochrome C and 7 molecules of APAF1 combine to form something called the apoptosome, or the wheel of death, which then cleaves the procaspase 9, which is a proenzyme, and once it's cleaved, it becomes caspase 9, which is the active form, basically. It's called an initiator caspase. And what the caspase 9 does is it cleaves the procaspase 3, which then turns into caspase 3, the active form. And once the caspase 3 becomes active, it has hundreds of substrates, and these are both proteases, by the way. So it has hundreds of substrates, thousands of substrates. It destroys the cell, basically, and it also cleaves something called PARP, which is a DNA repair protein. So they don't want the DNA to be repaired anymore. So you don't want this PARP protein to be active. That's why it's cleaved, basically. So over here we have, we can see in the cell that APAF1 is upregulated, and again, when we have the combination, as opposed to just each of them alone, there's a much higher concentration. So this shows that APAF1, which helps in apoptosis by forming the apoptosome, it's highly upregulated because of the combination more so than the individual one. So this shows, again, the combination has a much higher potent apoptosis pathway activation rather than just each of the products alone. Same for cleaved caspase 9. We can see the bottom line indicates cleaved caspase 9. We can see it's slightly increasing, and it's much more in the combination group, whereas regular pro-caspase 9 is less. That's the top line. We can see it getting lower. And same thing for cleaved caspase 3. Look at that. There's so much cleaved caspase 3 in the combination group, whereas there's almost none when sorafenib is alone or resveratrol is alone. Same thing for cleaved PARP. So there's much more cleaved inactive PARP. Or DNA repair protein, which you don't want anyway during apoptosis. You don't want it to be active. There's much more of the inactive form when you have the combination. So we can see here that through the actions of all of these proteins that the combination is actually increasing the apoptotic pathway much more than just each of these products or the drug and the natural antioxidant alone. So that's basically what this paper was saying. How sorafenib and resveratrol together, the synergistic effect was greater than if they did if each of them worked alone. And so basically they activated the ROS. The ROS was there's much more intracellular ROS, and there's P53, which activated P53, which then transcripted BAX, which then allowed cytochrome C to exit, go to the join with the APAP1, cleave caspase 9, cleave caspase 3, and lead to apoptosis. So that's basically the gist of the paper, how the synergistic effect of these two products increases the cytotoxic or apoptosis of cancer cells in the MHC7 line. Thank you.